Hello, I'm Jing Zhang from McGill University.
I'm Melanie Marshall from the University of West Indies in Barbados.
And I'm Angelica Bojo from McGill University. And we will be presenting, in collaboration with the National Conservation Commission, on optimal propagation conditions for pink, red, yellow, and white Mussaenda cultivars in Barbados.
The goal of our study is to test different wood types in Mussaenda as well as different methods of enhancing rooting success
in order to determine the best conditions for propagation individual to the 4 cultivars at study.
Our project was conducted at the National Conservation Commission, which is a government organization under the Ministry of Environment, Water Resources, and Drainage. The Commercial Department of NCC is responsible for the management of the Commission's plant nursery, garden center, grass nursery, and provides landscape services for both public and private sector. Our work was mostly done in the plant nursery, which covers an area of approximately 50,000 square feet with 4 different sections: a plant propagation area, a hardening area, potting area, and a holding area.
Donna aurerea is the parent of almost all Mussaenda cultivars and is characterized by abundant white floral sepals, often 5 per flower, and an orangey-yellow flower. Donna luz was created by the hybridization of Donna aurerea to Mussaenda erythrophylla. Their resulting progeny were self-pollinated and back-crossed to both parents, which led to the common pink edged red, greatly enlarged, and pendant sepals. Mussaenda erythrophylla is a species with red calyx lobes and 5-lobed tubular white to cream corollas with red felt-like centers native to tropical Africa. Mussaenda flava is no longer considered part of the Mussaenda genus, hence its name, though it shares many traits with the genus. Branched in terminal trichotomous corymbs, the bright yellow flowers, having a single light yellow to white enlarged calyx lobe per bloom, produce a faint perfume and bloom all year round in the tropics and subtropics.
For our project, growth medium was one of the variables tested. The 2 we used was sand and a peat mixture. The medium originally in the bench at the NCC nursery was sand. A rake was used to till this before planting. Propagation bench 8-3 was requested for use only when the sandy medium area of G1 was completely occupied. In order to prepare the bench with the peat mix, part of propagation bench G1 was completely removed of sand to be filled with our mixture. The growth medium was prepared with 70% peat, 28 shovels, 20% compost, 8 shovels, and 10% perlite, 4 shovels, for a total of 40 shovels. A wheelbarrow served as transportation from the preparation area to propagation bench and vice versa.
With the help of UWI Professor Jeff Chandler and McGill Professor Danielle Donnelly, Mussaenda Donna luz cuttings were taken on Monday, June 15th from the University of the West Indies Cave Hill campus. Mussaenda erythrophylla cuttings were taken on Thursday, June 25th from the flower forest. Mussaenda Donna aurerea and Mussaenda flava were finally taken on Friday, June 26th from Andromeda Botanical Gardens. Cuttings were taken from large branches while paying particular attention to encompass as much wood types as possible from basal, midshoot, and terminal positions. The base of the cuttings were wrapped in wet paper towel for hydration purposes. Cuttings were then placed in large blue plastic bags to protect and preserve moisture after being minimally trimmed to constrict volume and facilitate transportation. The enclosed cuttings were then placed into the cargo area of Professor Jeff Chandler's pickup truck and rushed back to the NCC for propagation. Once again, limiting the time between cutting and planting is an extremely important factor in ensuring rapid and successful rooting. On arrival at the NCC, a disinfected bucket was filled with a diluted bleach mix, 10% Clorox, and another was simply filled with water, as a method of disinfection and sterilization to remove and rinse away most fungi, insects, or other pests that may have contaminated the plant. Similarly, the secateurs used in the cuttings were disinfected by cotton swabs dipped into surgical alcohol at an interval of approximately 8 cuttings. Following an efficient cutting process with effectively distributed division of tasks, the following was done in the least amount of time possible with the utmost precision and diligence.
Okay. When doing cuttings, it's really important to cut just below the node and in the same direction as the node, in a slant. Like this. Then We are actually doing our cuttings based on 3 to max 4 nodes. So we have to see how long they are because each have to have approximately the same length and the same width.
The bud or the flower buds in softwood cuttings were cut off straight at the top to reduce stress. The cuttings were then trimmed to retain a maximum of 2 pairs of leaves per stem cutting on the upper portion. Finally, the cuttings were dipped into the bleach solution, rinsed in water, and then laid out on a clean bag to dry one by one. Subsequently, basal wounding was performed with the help of a pruning knife on the basal portion of non-softwood cuttings, controlling for basal wounding effects, 20 semi-hardwood and 20 hardwood for each cultivar. To maximize cambium exposure, straight lines were cut vertically down the base of the cutting.
So now we are presently heading to our bench to plant the cuttings we just took. These ones are the yellow Mussaenda ones. Now we are dipping our cuttings into the rooting medium. So it is really important that we dip just the base and then top off the excess for those that aren't wounded. But for those that have been wounded, we have to take our cutting, dip it into the rooting medium, and make sure to tap a bit of the powder on the wounded areas. This way, the rooting powder can infiltrate into the cambium and help the plant grow even more.
Weekly measurements were taken to gradually determine growth indicators and compare success of the different variables at study based on growth. The number of nodes, number of leaves, and leaf diameter was measured during a 6-week interval. Buds were not counted as leaves until they reached a diameter of 2 centimeters. A sample was selected for each group, marked, and consistently analyzed to average conclusions. Number of nodes were counting above ground, excluding 1 to 2 nodes below the ground. Overall performance and additional observation notes were taken to compare germination results for control, rooting powder, and rooting powder with basal wounding variables in the peat growth medium to those in the sand. As Mussaenda is known as a difficult-to-root species, it is critical not to transplant the cuttings too early into the hardening area. We carefully remove the cuttings from the propagation growth medium on week 6, 42 days after planting, and the rooting results were determined at this stage. Besides the number of cuttings rooted in each group, we recorded the length of the longest root and the abundance of adventitious roots for comparisons of rooting performances. For pink Mussaenda, a total of 45 cuttings were rooted. The softwood cuttings with rooting powder that grew in the stand medium had the highest rooting rate of 80%, and the roots were as long as 7 centimeters. For red Mussaenda, a total of 20 cuttings were rooted. The best group was also softwood with rooting powder in the stand medium with a success rate of 40%. For yellow Mussaenda, a total of 43 cuttings were rooted. The cuttings grew and rooted better in the peat medium, and the group of softwood cuttings with rooting powder achieved a very high success rate of 80%. The hardwood cuttings had great rooting results in peat as well, and rooting powder in combination with basal wounding treatments proved to have positive effects on rootings. For white Mussaenda, only 4 cuttings were rooted. Although quite difficult to determine due to the excessive desiccation rate for this group, our results suggest softwood control cuttings in peat treated with rooting powder alone to be the best group for propagation, with a 20% success rate. This is quite common for hardest to root cultivars. The cuttings were then taken to the preparation area and transplanted into potting bags. The plastic potting bags were filled with the soil compost medium commonly used at the NCC, and 2 to 3 cuttings were planted into each bag. The plants were watered and then transported to the hardening area. Once they have been fully acclimatized after about 4 weeks, the best specimens will be given to the donors of the cuttings in recognition of their contributions. For future propagations at the NCC, we recommend to take softwood cuttings and treat with 0.3% IBA rooting powder before planting in sand for both the red and pink Mussaenda. For the yellow Mussaenda, the growth medium should be changed from a sand medium to a peat-based mixture, but still use softwood cuttings and apply rooting powder. As for the hardest to root white Mussaenda, we suggest mound layering as an alternative propagation method to raise the survival rates of cuttings. From our experience, we also learned that it's important to select healthy plant tissue sources to avoid fungi contamination in propagation. Slug and snails did occur but could be easily controlled with methaldehyde pellets, the pesticide commonly used by the NCC. While at the NCC, we got the chance to visit the brand new tissue culture lab, which is used for the micropropagation of several plant species. We were fortunate enough to be able to actually assist and participate in the propagation of the white Mussaenda on a smaller scale than the method our research focused on. A number of specimens were presented to us, demonstrating the different stages into the process of micropropagation. This was quite fascinating to witness, and the technology used was impressively advanced for the size of the facility. The lab was very sterile, and therefore shoes had to be removed at the door. Feet and hair were to be covered with a hairnet once inside. There are several steps associated with disinfecting the specimens before the actual procedure could begin. This included the use of bleach, dishwashing liquid, and distilled water, precisely requiring up to 15 minutes of immersion. After the specimens have been adequately disinfected, the micropropagation process is then carried out inside a laminar flow hood. This is done under strictly aseptic conditions. Several precautions should be taken, including ensuring horizontal transmission of materials, as crossing hands increases risk of contamination, and therefore all tools must move from left to right or right to left. Similarly, it must also be ensured that the hands do not pass directly over the tile on which the specimen is placed, as another preventative measure against contamination. All tools are stored in alcohol and must first be transferred into the distilled water before being used to handle the specimen. It is then placed back into the alcohol after final use. The cultured specimen is removed from the test jar with sterilized tweezers. Scissors are then used to carefully cut off 4 sides of the Mussaenda leaf to induce callousing. The tweezers are again used to place the cut leaf into a petri jar containing agar and other ingredients conducive to growth. After the specimen is placed in the jar, it must be labeled and transferred into the holding room. The tile is removed and the entire area is thoroughly cleaned and disinfected, especially the operation tile, for the next procedure.
We would like to thank James Seeley for believing in our research and trusting that our experiment will be fruitful.
And Sean Phillips for getting us through different processes, providing emergency material, and recommending procedure based on experience.
And of course, the many NCC staff for their warm welcoming and greatly appreciated assistance.